advertisement. Yes, at 24th you joined class and we did biomolecule, primary metabolite, sugar, protein and fat, right? Am I correct?
Yes, sir.
Okay, fine. After that, 29th, it was a leave, right? We did not take in any class. And again, we started with the 1st of December. In 1st of the December, we did enzyme and reactions and nucleic acid. Three things we have done. And for sake of enzyme, I have given a PDF to Niskarchi. So what you have to do after the class, you have to take that PDF from Niskarchi. That is actually my handwritten notes, and everything is mentioned over there. If you'll go through with these notes, you will definitely come to know what exactly is it. I have just explained those notes only. And in case if you are facing any problem, so just discuss me in next class. And one more important thing, like in the same class, I have covered this one. I have covered nucleic acid. So that is really very important. I have told what are the sugars, pentose sugars. Second, I have told what are the nitrogenous bases. Again, what is the structure of DNA and RNA, right? What is nucleotide, nucleoside. I have mentioned all these things over there in that video. So what you have to do, take the backup of last class, fine, that I have taken on the 1st of December. Go through with that. And nucleic acid, I think I have mentioned in, like I have discussed in 20 minutes. So you have to take that video for 20 minutes, and rest of the part I have covered that complete notes, right? So two things you have to do. In case if you are uncomfortable or have any query, any question, feeling any problem, so you can contact me in next class, right? Next class we can discuss that. Is it clear?
Yes, sir.
Okay. So what should start? What should be the next impact? So we need to discuss what we did. So we have completed, I think we have completed the unit 3 as well. Am I correct, Maimuna?
Yes, sir.
So should we start with the plant physiology or human physiology? Tell me.
I think we can go with plant.
Plant physiology, fine. So for the plant physiology, we have to start with the transport in the plant, right? So we have to start with this chapter only, transport in plant. Okay. So it's easy, but need to understand lots of the things, not a smaller one, big one. Mauna, did we have covered unit 2? We have covered unit 2 now?
I think we didn't cover it, sir. We just went to unit 3 because we were having exams.
Exactly. So what should do? First, go through with unit 2 or unit or plant physiology. Tell me.
After unit 4, we can go to unit 2.
Okay, so first we should start with the plant physiology, right?
Yes, sir.
Fine. So start with the plant physiology. No issue.
I also got the marks also for this test, which I did.
Okay, so how much marks you got?
I got 23 out of 25.
23 out of that's good score. Great, very good. You are doing well. Good. So was it offline or online exam?
Offline exams. Your schools are open now.
Okay, schools are open over there. We are again getting the sign to get these closed down. Many part. Transport in plant. Yes. So start with the transport in plant. So transport in plant means we are going to study two things. First, that is a transport of food, and second, that is a transport of water. So before going to the transport of food and transport of the water, we have to discuss like the different type of the channels, different type of the proteins, and what is the diffusion, what is the osmosis. So these are the basic concept, but they are very tricky kind of the concept. So let's start with the transport part. So first, there is a topic which is water potential and all that you need to take care of. The rest of the things are Easy. Fine. So let's start with the diagram. Let me open a diagram. Plant physiology. Active transport, passive transport. Right. So first we will start with the transport of substance inside and outside of cell, right? So we'll discuss the type of transport. Okay. So first, Mamuna, we are going to discuss what is the active transport, what is the passive transport. So active transport are the transport where we have to spend energy, right? So put the heading means of transport. Heading is means of transport. First, look, Mamuna, what we are observing over here, this is actually the plasma membrane. And you can observe it is made up of, as we have already understood complete cell biology. So as you can observe over here, this is the one layer of fat, this is the another layer of fat. So this is the actually plasma membrane or biomembrane. So this is plasma membrane. Plasma membrane, we know that the maximum part of the plasma membrane is made up of bilayer of lipid, and some of the part is made up of protein. And I have told you that there are some channel protein or gateway protein. Okay. So those substances which are soluble with the fat, they can come inside through the process of diffusion. And you know very well that diffusion takes place across the membrane from higher gradient, like higher concentration, to lower concentration. So fat soluble substances, they just cross these plasma membrane from the fat region or from the fat bilayer or lipid bilayer, right? So they just come by this way. But those substances which are not fat soluble, like they are hydrophilic. So these substances, fat soluble substances, we can say the transport of hydrophobic, transport of hydrophobic means which are fat loving substances or which are fat soluble substances. They are transported by means of the passive transport, but transport takes place through fat bilayer, simply by the process of diffusion. Again, what is facilitated diffusion? Look, ma'am, what is diffusion and what is, can you tell me what is a facilitated diffusion? Facilitated. Look, whenever a school bus comes at your school, suppose that you are at a school and your school bus is going to drop you at your home. So what you will do, like the concentration of the, like the number of the students are more outside and inside the bus is vacant. What will happen? All the those students who are in 11th, 9th, 10th and 11th, they will go inside the bus and bus will become full. Whenever the bus will become full, that the student will stop going inside the bus and they will drop at your home. And so this is one thing, that is the diffusion process. What is facilitated diffusion? Whenever bus arrives at school to pick those students who belongs to kg1, kg2, nursery and all those, like they are very small kids, they cannot step up inside the bus. Even there is the bus is vacant and the more students are outside. So what will happen? There will be a helper. What this helper will do? This helper will pick the hand or hold the hand of a child and it will pick inside him, right? So that's how they will pick the child, right? So they are helping. That is called actually facilitated diffusion. So there is a person who is not taking the money, but it is or he's providing the facility to do all these small kids. He's helping them out, right? So that is a facilitated diffusion. So in the same way, for transport of hydrophilic substances, which are water loving substances, hydrophilic. So hydrophilic substances, they are not fat soluble, they are water soluble. They cannot cross the plasma membrane from this lipid region. They have to cross through with the help of these channel protein, right? They are channel protein. We have already discussed the channel protein. Mamuna, did you remember? We have already discussed channel protein.
Yes, sir.
Yes. So these are the intrinsic protein. They will cross through it from higher concentration to lower concentration, right? So this is the facilitated diffusion, right? What is the active transport? Generally, active transport takes place against the concentration gradient, right? So here the concentration, again you can see here these are the five particles, here these are the two particle only. So what happens when the transport takes place? So let me give you example. Suppose that uh, protoplasm have higher concentration of sodium and external environment have lower concentration of sodium, but still if the protoplasm requires sodium, it will spend energy, and at the expense of energy they take material inside. This is called active transport. Is it clear, Mauna?
Yes, sir.
Okay, so please note it down. What is active transport? What is passive transport? So first, diffusion. First heading is diffusion. Movement by diffusion is passive and slow. Full stop. It occurs along the concentration gradient, means from higher to lower concentration. It occurs along the concentration gradient, that is from higher to lower concentration. That is from higher to lower concentration. Full stop. Provided the plasma membrane is permeable to this substance. Provided plasma membrane is permeable to this substance. Full stop. Next point. Facilitated diffusion. The particle which are water soluble cannot pass through plasma membrane, easily. Full stop. The hydrophilic solute pass through the protein channel. Full stop. For facilitated diffusion, the membrane possesses aquaporin. This question have been asked in NEET exams. What is the aquaporin? Aquaporin and ion channel. So look, Mamuna, these are the ion channel and aquaporin. Aquaporin are responsible for actually transport of the water. So they are a type of intrinsic protein and they are responsible for passage of the water. Ion channel, they are responsible for the transport of the chain. Both help in facilitated diffusion. Clear?
Yes, sir.
Aquaporin, note it down in the same point. Aquaporin are water channel and they help in passive transport of aquaporin are water channel and they help in passive transport of water soluble substances. One more point. In facilitated diffusion, energy expenditure do not require. In facilitated diffusion, energy expenditure is not required as they takes place through the concentration gradient. This diffusion, one more important one, this diffusion allow very specific substances. Okay. Now, in the same point, like I'm not going to mention here active transport. First, I will elaborate more passive transport. Mamuna, is it clear? Passive transport, like if diffusion and facilitated diffusion, is it clear?
Yes, sir.
Okay. I'll take the screenshot and I will tell you one more diagram. I will show you one more diagram and explain some more points.
Done, sir.
Okay. Now, what are the actually channel protein? I want to show what are the channel protein actually, and what are the type of channel protein, right? So again, the channel protein can be of different types. Actually, these are the channel protein. So what happened? Channel protein may be uniport. So what are the uniport? Uniport are those substances. Uniport are those port which allow of only one type of the substance in only one direction, right? So this is the uniport. What happened in symport? Two ions are transported along with each other, right? So there are two types, uniport and co-transport. In co-transport, two ions are transported simultaneously. In uniport, only one substance is transported. In co-transport, two ions are transported simultaneously. If they are in same direction, this is called symport. If they are in opposite direction, this is called antiport. For sake of example, let me tell you antiport. So in antiport, whenever the in neurons, the membrane of neurons, what happen? When sodium will come inside, potassium will go outside. So both are the positive ends. When sodium will come inside, then potassium will go outside. If potassium will go inside, then the sodium will go outside. So this is the antiport. So they have antiport. Symport, Sometime what happened, if calcium is coming inside, then sodium will also come inside. So that is the this sodium potassium pump is antipode, and that's how the co-transport takes place. Is it clear?
Yes, sir.
Okay. So please note it down. There are two major type of transport protein. That is carrier protein and channel protein. So these are the actually channel protein. Carrier protein and channel protein. Full stop. Carrier protein. Look, Mamuna. So what is the carrier protein? Carrier protein. So the person who is holding the hand of kid and like bringing him inside the bringing him or her inside the bus. So that person is that gate is actually channel, and that person is carrier protein. Are you getting my point?
Yes, sir.
That gate of the bus is channel, and the person who is standing and bringing him inside, that is the carrier protein, right? So same thing happens. Channel. I will show you one more diagram of channel protein. So note it down. The carrier protein bind particular solute. Carrier protein bind the particular solute and transport and deliver it. Transport and deliver it the other side of membrane. Full stop. The carrier protein, sorry, some carrier protein, next point, some carrier protein allow transport only if two type of molecule moves together. Full stop. This is called co-transport. Full stop. Co-transport may be symport or antiport. Co-transport may be symport or antiport. Full stop. Next point. In symport, two ions goes in same direction, while in antiport both of the ions move in opposite direction. One last point. When a molecule moves across membrane, when a molecule moves across membrane, independently, it is called uniport. Clear, Memuna? Co-transport, symport,
Yes, sir.
antiport, uniport, facilitated diffusion, and diffusion. Right. Now I want to show you what is the this one carrier protein. Right. So let me show you the what is the carrier protein. Okay. Okay. Yeah. Look. Uh, look. So how does the carrier protein work? Hydrophobic substances like fat loving substances, they come through this side. The carrier, this is the channel. This is channel protein. This is closed. Uh, like there is, this is the some stimulus molecule. It may be hormone. It may be anything. This is binding site. It bind. When this bind, after the binding, what happened? The channel protein binding of a stimulus molecule causes the pore to open. This channel will open. Now the hydrophilic substance will come down. So this is the, that's how the carrier protein actually work. Clear? Channel protein, they always remains open, right? But particular type of substances can pass through them. But channel protein, they changes actually their carrier protein, they changes their shape. So due to the changes, in their shape, their gate open and this gate closes down, right? Is it clear, Mamuna? Carrier protein?
Yes, sir.
If you want, you can take the screenshot of this PPT. Taken? Okay.
Yes.
So now next heading is active transport. Active transport. Active, we know that in active transport, as we have just discussed in this diagram, first one. So in active transport, the expansion of ATP will take place, and then the only gate will open when the ATP will expand. Then the gate will open, and the substance will come inside. So please note it down. Active transport, the movable carrier protein called pump. Here, these carrier proteins are known as pump. So in active transport, we call these carrier proteins pump. In active transport, the movable carrier protein called pumps, they utilize ATP energy. They utilize ATP energy. They utilize ATP energy for the transport of substances. For the transport of substances across the membrane. Full stop. They require energy because transport of substances, they require energy because transport of substances takes place against concentration gradient. Transport of substances takes place against concentration gradient. Clear?
Yes, sir.
Carrier proteins are highly specific. Please underline this statement. This is a question for medical. Carrier proteins are highly specific like enzymes. Means there is a specific carrier protein for a specific ion or a specific substance. Okay. So this was the means of transportation, transport, how the transport takes place, active transport, passive transport, what are the aquaporins, right? What are the pump? Okay. Now the next heading is plant-water relationship. So in plant-water relationship, first there is a term, it's important for your school exams. What is the imbibition? Imbibition. So in plant-water relation, the first thing is, can you tell me, Amina, what is imbibition? Look, Mamuna, if you fill a bottle with grams, dry grams, put water inside it and fill the bottle tightly and put water inside. What will happen? Gram will break the bottle because of their pressure. So grams are very dry and hard kind of substance, solid kind of substance. When solid substances take water inside or absorb water, this phenomena is called imbibition. Clear?
Yes, sir.
Okay, note it down. The absorption of water by solid particle of an absorbent. Absorption of water by solid particle of an absorbent, causing it to enormously increase the volume, causing it enormously increase the volume without forming solution, is called imbibition. Without forming solution is called imbibition. Example, dry seed, dry wood. You can see during the rainy season what happened, your wooden window, it become hard. Sometimes it is really difficult to close it down. What happened? They absorb the moisture from atmosphere and they become very hard and very tight. So that is also an example of imbibition, right? Diffusion, we have studies. So again, what is diffusion? Second point is diffusion. We'll study the things one by one. Maimuna, diffusion, simple. What is diffusion, Maimuna?
It is something which has it is a spreading light from a light source evenly to reduce harsh shadows.
Simple, Maimuna. Look, what is that? You have a deodorant bottle, and the number of the molecule of deodorant are have high concentration inside the deodorant bottle. Whenever you push that deodorant bottle, what happened? These the molecule of deodorant start coming from higher pressure to lower pressure, right? Right. So that is the
Yes.
diffusion. So diffusion is simple. Example, for sake of example. So this is a there is a this is a wall, and here it's a hole. Right? Gas, liquid, whatever the substances are over here, the concentration of molecule is more over here, right? So concentration of molecule is more over here, and concentration of molecule is less over here. What will happen? Molecule will starts moving from higher concentration to lower concentration. Up to what extent? When the concentration becomes equally at both of the side, right? So this is high concentration. Here it's low concentration. So molecule will start moving from here to here, from higher concentration to low concentration. When the concentration will become equal at the both of the side, we will call this situation equilibrium. Equilibrium. So when they will achieve equilibrium, this process will stop. That is called diffusion. Clear?
Yes.
Okay. So please note it down. The tendency of part of molecule, atom, or ion, etc. The tendency of part of molecule, atom, ions, etc. of gases, liquid, or solid. Of gases, liquid, or solid. To get evenly distributed throughout the available space on account of kinetic motion is called diffusion. Full stop. In diffusion, the molecule moves from higher concentration. Molecule will move from higher concentration to lower concentration. That is concentration gradient. That is concentration gradient. Full stop. Now, what is the diffusion pressure? dP. So this is the diffusion. Diffusion clear, Amuna?
Yes, sir.
Okay. Look. So whenever suppose that you have a deodorant bottle. So this is a deodorant bottle and this is your room. Whenever you open the deodorant bottle or whenever you open the your gas cylinder, what happened? Gas starts coming from higher concentration to lower concentration. If you put the hand in front of the mouth of or in front of that pore of the deodorant bottle, so you will feel the pressure. Isn't that? You'll feel the pressure.
Yes.
Yeah. So that pressure is called diffusion pressure, and we use a term dP. So whenever the molecule move from higher concentration to lower concentration, they impart pressure, and this pressure is called diffusion pressure. dP. Put the heading dP. Diffusion pressure. dP. The pressure exerted by particle. The pressure exerted by particle to diffuse from area of higher concentration to lower concentration. Area of higher concentration to lower concentration is called diffusing pressure. Full stop. Now look, Mamuna, if the difference between the pressure is more between these two, high and low, then dP will be very high. If the diffusion pressure is very like if the diffusion pressure like if the concentration difference is less, then the diffusion pressure will be less, right? If the concentration difference is more, then diffusion pressure will be more. If concentration difference is less, then diffusion pressure will be less, right? So look, write it down. dP is proportional to dP is proportional to concentration of diffusing particle, right? So this is the dP. Now what is the osmosis? Mamuna, tell me what is osmosis. Clear till here? Mamuna, is it clear till this point?
Yes.
Okay. Now tell me
Sir,
what is
once
the
again I'll draw the diagram.
which one?
This
Okay. You want
molecule.
to? Okay, fine, fine. Please note it down. No issue.
Done.
Okay. Now what is osmosis?
It is a process where a solvent passes from a solution of low concentration to a solution of high concentration.
Great, great. That is called osmosis. Fine. So osmosis. Look, this is the diagram of osmosis. So what happened here? The water molecules are more, and here the water molecules are less, and sucrose molecule are more. Right. So what happened? The movement of but provided there should be a differently permeable membrane, which is also known as semi-permeable. Look, semi-permeable membrane have very small pore. It do not allow the passage of these sugar molecule or these larger molecule. It allow the passage of only water molecule or solvent molecule or the molecule of small size. Generally, they are the solvent molecule. So through this solvent molecule only can cross, right? What will happen? They will start flowing from higher concentration to their lower concentration. So solvent molecule will pass through The only difference between the diffusion and here the osmosis is that the semi-permeable, right? So this is called osmosis, right? Please note it down. Diffusion of water, diffusion of water from its pure state or dilute solution. Diffusion of water from its pure state or dilute solution into a solution or stronger solution, into a solution or a stronger solution when two are separated by semi-permeable membrane, when two are separated by semi-permeable membrane, that is termed osmosis. Right? So this is the definition of osmosis, and this is the process of osmosis. And look, the osmosis can be of two types. You'll get the question from this one: endosmosis and exosmosis. So osmosis, note it down. Osmosis is of two types: endosmosis and exosmosis. Can you tell me what is the difference between two, endosmosis and exosmosis?
Osmosis means the procedure takes place inside the body or inside the
For
object, and
example,
exosmosis.
for sake of example, look, I will tell you, then I will ask one more question. Look, Memona, what happens? Suppose that you have a glass and you have put pure water, like mineral water, like simple water, over here, and put a resin over here. What will happen? After some time, the water will go inside and resin will swell. Yes, that will happen.
Yes.
After some time, the resin will go swell, right? And that's how happen. You put almond in any water bottle. So what happened? The water comes inside the almond, they swell, and this is called endosmosis. So when the water is going inside the system, this is endosmosis. Now have a different experiment. We have another glass. What we did in this glass, we have salt concentration, right? So one more thing I want to add on over here, Mamuna. The outer side concentration is low, right? Low concentration. And inside the resin, they have high concentration. High concentration. Again, we use one more term. Low concentrated solution is called hypotonic, right? And high concentrated solution is called hypertonic, right? So movement of water will take place from hypotonic to hypertonic solution, and resin will swell. This is called exosmosis. Again, what happens? What you are doing over here, you have a salt solution over here. This is salt solution. Means high concentration solution. Means hypertonic. So external medium is hypertonic. That is salt solution. So this is hypertonic solution. What will happen? This is a grape molecule. This is a grape. Now, when you will put the grape in, you will put the grape in salt solution. Inside the grape, the solution is what kind of solution? Hypotonic. What will happen? You will see after some time, the water will start coming out from the grape, and the grape will shrink. So when the water come outside from the system, this is called exosmosis. Clear? This is the best example. Mamuna, is it clear? Exosmosis, endosmosis. Clear?
Yes, sir.
I have a question for you. I have a fish, and the fish is belongs to this fish belongs to uh, like a river fish. It's a freshwater fish, right? What I will do, I will put some marine water and put this fish inside a marine water. What will happen? Means I have picked a fish, a living fish from soft water or freshwater pond or freshwater river, and I have put this fish in marine water. What will happen in oceanic water?
I think they will lose the water from their bodies due to hypotonic environment.
Right, very good. And the fish will die. So technically, exosmosis will take place, and what fish will start losing water from its body, and the fish will die. Right? Same. Like these are the questions. That's how they ask the question. So same time, you can say if you pick a scoliodon, a freshwater like or a marine fish, the concentration of the blood of that fish will be higher. And if you'll put this fish in a freshwater solution, so what will happen? This water will start going inside the fish, and due to the gushing of the water means endosmosis will take place, and that marine fish will also die. Right? So please note it down. Endosmosis, the osmotic entry of water, takes place into the cell, organ, or system. In endosmosis, osmotic entry of water takes place into cell, organ, or system. Full stop. For example, if we put a resin in fresh water, for example, if we put a resin in fresh water, resin swell after some time. Next, exosmosis. In exosmosis, osmotic withdrawal of water from cell to organ or system, from cell to organ or system takes place. Clear?
Yes, sir.
Okay. So if the both side concentration will become clear, become the same, that will call isotonic solution. Isotonic. So isotonic solutions are those where the concentration is equal at both of the sides. Hypertonic higher concentration and hypotonic lower concentration. Okay. Osmosis, yes. Okay. Now, what is the DPD? And before going into DPD, we need to discuss some terms. Turgor pressure, wall pressure. Okay. Take a screenshot. Emana? Let me show you a diagram and let me make you understand. Look. What happened, Memuna? When this is a cell, typical a plant cell, this white part belongs to vacuole. Outer layer is cell wall, inner is plasma membrane. This green part is protoplasm. What will happen when water will coming inside the cell? Water moves into cell due to osmotic concentration of cell content. So when the water move from outside to inside, so this water will impart osmotic pressure, OP. Clear? We have discussed few minutes before that what is the OP, osmotic pressure. So osmotic pressure, it is exerted due to the movement of solvent molecule inside or outside the system. This is called osmotic pressure. So when this water will move inside the cell, like not cell, inside the vacuole, in fact, technically inside the vacuole. So movement of water will take place from outside to inside the cell in the vacuole. This impart a pressure. This is called osmotic pressure. So osmotic pressure will be imparted from outside to inside the cell. Again, what will happen? Now the vacuole will become enlarged and this cell will become turgid. Due to this, due to turgidity, because it has become swollen due to the turgidity, right? So due to turgidity, what will happen, Mamuna? This vacuole will put a pressure from inside to outside, from inside to outside of the cell. This is called TP. So osmotic pressure is inside the cell and TP, it opposite pressure, TP, target pressure, right? Target pressure is imparted by the cell towards the outside on the cell wall due to turgidity. Again, to balance it, this cell wall will put pressure inside. That is called wall pressure, WP. So against the TP, the cell wall will put a pressure called wall pressure, that is called against TP, right? So cell content press the wall, so that is called target pressure. Cell wall retain expansion of the cell with wall pressure. This is called wall pressure, WP. So this is WP, this is TP, and this is OP. Is it clear or not, Mamuna?
Yes.
Take this screenshot. Now I want to tell you what is the DPD, diffusion pressure deficit. So my topic is diffusion pressure deficit. What is DPD? Okay. So put the heading DPD. What is OP, TP, and WP? It is mentioned already over here, so I need not to mention DPD. Diffusion pressure deficit. Diffusion pressure deficit, right? Can you tell me, Mamona, what is the diffusion pressure deficit? Look, Mamona. Diffusion pressure. So pressure V is always measured in bar, right? So suppose that here is pressure 10 bar, right? And here is pressure 5 bar. So what is the deficit? The deficit is of 5 bar, right? Clear? Clear, Mamona, or not?
Yes, sir.
So when the pressure will become at both of the side 7.5, 7.5 bar, the osmosis will stop, right? So this is called DPD, diffusion pressure deficit, because here is deficiency of 2.5 bar, right? In fact 5 bar, right? Put it down. The pure water have maximum diffusion pressure. Let's stop. Its diffusion pressure gets lowered by addition of solute particle addition of solute particle in it. Is it clear? If we'll add the solute particle, the concentration will become higher and more water will move inside it, right? So if solvent is added, solute is added, the dpd will decrease. The reduction is diffusion pressure. The reduction is diffusion pressure of water in a solution over pure state, in solution over pure state is called dpd. Full stop. The dpd is also called suction pressure. dpd is also called suction pressure. Look, ma'am, what is I want to tell you. Suppose that, so actually the diffusion pressure is pure water is maximum, right? So suppose that for sake of example, diffusion pressure of pure water, both of the side we have pure water, right? Diffusion pressure is maximum. This is one. This is one. What will happen? Both side there is a pure water, and what I am doing, I am adding sugar over here. Sugar over here. So I have added sugar, right? After addition of sugar, what will happen? The diffusion pressure will decrease, right? So suppose that it become 0.7. So how much dpd is there? Mamuna? Deficit is of 0.3. Clear or not? Please tell me.
Yes, sir.
So that is a deficit, means deficiency, right? Shortness, right? So dpd addition due to addition of the sugar or due to addition of any salt in any pure water, the dpd will decrease, right? Okay. So again, so now let's discuss this term in technical way, more technical. What is the water potential? What is water potential? So take the screenshot and then move ahead. I'll tell you what is the water potential. Next heading is water potential. Again, I want to give you a formula. dpd is equal to osmotic pressure minus wall pressure, or we can say wall pressure is equal to turgor pressure. Both will be equal, right? So you can, in formula, you can write it down. dpd is equal to op minus wp, or even you can say op minus tp. Both of this. Okay, Mamina. Take the screenshot or write it down, then let me know.
Done.
Now I am going to tell you what is the water potential. Water potential. Let me explain. Look, Mamuna, actually pure water have the maximum water potential. So what is the water potential? Water potential is the kinetic energy, free kinetic energy, right? So the pure water have maximum water potential, and we represent water potential with the help of a symbol called psi. So this is a symbol called psi. So water potential, water potential. So water potential is represented by a symbol called psi W. It means water potential. What is the water potential? Water potential is actually free kinetic energy of water. Free kinetic energy of water. So the pure water have water potential is equal to 0, right? So here the water potential is 0. If you will water potential is 0. If you will put some solute particle into it, what will happen? Water potential will come down. So 0, it will become what? Negative. Clear? Right? So
Yes.
now we are like nowadays we are representing the osmosis in terms of physics, right? So in terms of physics, if you will say, in terms of chemistry, we discussed the movement of molecule takes place from higher solvent concentration to lower solvent concentration. The same thing we are representing. This is the same thing only. We are just explaining it in the from the point of view of physics here. So we can say in osmosis, the movement of water always takes from higher water potential to lower water potential, means from 0 towards the negative. Is it correct or not?
Yes.
Note it down. Water potential. This is the chemical potential of pure water. This is the chemical potential of pure water at normal temperature and pressure. Normal temperature and pressure, which is always 0. Full stop. Water potential is actually the free energy of water, free energy of water in any system. Full stop. In solution, the value of water potential in solution, the value of water potential is always negative, or less than 0. Always negative or less than 0. Full stop. The water potential is represented by a Greek symbol psi W. Now the next point have been asked in your medical examination. What is the unit to measure water potential? Right. Full stop. Next point. The value of water potentials is measured in bars, Pascal, or atmosphere. Bar, Pascal, or atmosphere. Now we can say in terms of water potential, movement of substance from area of movement of substances from area of higher free energy from area of higher free energy to lower free energy is called diffusion. Right. So we represent the water potential. Psi W is equal to, it is water potential of a solution, maybe psi W is equal to psi S plus psi P. What is psi S actually? Psi S is solute potential. Solute potential. Now, what is this solute potential? Let me explain you what is the solute potential first. Suppose that here the water potential of pure water was, for example, 0, and I have added some solute, and now the water potential is 0 point minus 0.3. So it have dropped 0.3, right? So this dropping of 0.3, it will be called solute potential, right? Note it down. Psi S, psi S represent the solute potential. Full stop. Solute potential is the effect of solute on free energy of water. Full stop. What is psi P? Psi P is called pressure potential. Pressure potential. So note it down. Pressure potential is effect of pressure. Pressure potential is effect of pressure on the free energy of water, and the formula is psi W is equal to psi S plus psi P. So water potential is always equal to solute potential. Water potential of a solution will be always equal to solute potential plus pressure potential. Is it correct? Is it clear?
Yes.
Okay. So this is the something. So we whenever we will talk right now nowadays, whenever we talk about the diffusion, we osmosis, we always talk about the water potential. Water potential means the free energy of water. So if the free energy of water, like pure water, have the maximum free energy, their molecules are free to move here and there. If you will mix the solute, their free energy will reduce because they will start colliding with the solid molecule, right? So their free energy decreases. So whenever nowadays we talk about the any water relation, we always use the term water potential. Clear?
Yes,
Okay.
sir.
One more concept I want to discuss is this one, plasmolysis. What is the plasmolysis? With the deplasmolysis, water potential we have discussed. Water potential, solute potential, osmosis. Put the heading plasmolysis. Now let's discuss the plasmolysis. What is the plasmolysis? Amana, in case if you are if you are unable to understand any concept, never hesitate.
Yes, sir.
Plasmolysis. So this is the process of plasma. Actually, this is the turgid cell, means fully swelling cell at normal state. So this is the actually turgid cell. What happened when uh, this is the normal turgid when the cell is placed at hypotonic solution? What will happen? In hypotonic, so inside the environment there is a hypertonic. Water will start going inside the cell and the cell will become turgid, swell. The cell is normally turgid condition. Vacuole will be full of the water and it will become turgid. So in hypotonic solution, the cell remains turgid. What happened in isotonic solution? In isotonic solution, the concentration is equal at the both of them, inside the cell and outside the cell. So some water will go inside and some water will come outside. So this is normal cell called flaccid cell. What happened when you lose the cell in hypertonic solution, the water will starts coming on out from the cells. That is called plasmolyte cell, right? So this is called plasmolysis, and this cell is called plasmolyte cell, right? So turgid cell. This is plasmolyte cell. There is one more situation: incipient plasmolysis and evident plasmolysis. This is actually this plasmolysis. This plasmolysis is called incipient plasmolysis, right? This is called incipient plasmolysis. And again, there is one more stage that is called evident plasmolysis. Evident plasmolysis. What will happen in evident plasmolysis? In evident plasmolysis, except one or two places, this protoplasm will leave the space, right? This protoplasm, this is the vacuole. This protoplasm will leave the cell wall. And here, the reason, so this is in very hypertonic solution or a strong hypertonic solution. The external solution will start filling in this area between the protoplasm and this cell wall space. This is external hypertonic solution that will start going inside. This is called evident plasmolysis, and this is incipient plasmolysis. Is it clear, Mamuna?
Yes, sir.
Take this screenshot, then write it down. Noted?
Yes.
This is incipient plasmolysis, and this is evident plasmolysis. So start noting now. First, put the heading plasmolysis. Shrinkage of protoplasm of cell, shrinkage of protoplast of a cell from its cell wall, from its cell wall under the influence of, from its cell wall under the influence of, under the influence of hypertonic solution under the influence of hypertonic solution is called under the influence of hypertonic solution is called plasmolysis is called plasmolysis. Next point. Hypertonic solution causes exosmosis. Hypertonic solution causes exosmosis or withdrawal of water from the cytoplasm and then central vacuole of cell, then central vacuole of cell. Full stop. The first stage of plasmolysis is called limiting plasmolysis. Limiting plasmolysis. So first stage of plasmolysis is called limiting plasmolysis. Right? Next point. Initially, the protoplasm withdraw itself. Initially, the protoplasm withdraw itself from the corners. Full stop. The stage is known as incipient plasmolysis. Full stop. The hypertonic solution now enters the cell in between protoplast and cell wall. The hypertonic solution now enters in cell between the protoplast and cell wall. Full stop. Due to continuous exosmosis, protoplast shrink. Full stop. The further withdrawal from the cell wall except one or few point it is called evident plasmolysis. Everything clear, Mamuna?
Yes, sir.
Now, what is deplasmolysis? So, reverse of plasmolysis is called deplasmolysis. Note it down. The swelling up of plasmolyzed protoplast under influence of hypotonic solution or water is called deplasmolysis. Full stop. It takes place. It takes place it takes place due to endosmosis. Please put a note. Deplasmolysis is possible only immediately after plasmolysis. Deplasmolysis is possible only immediate after plasmolysis. Else, the protoplasm will die. Else, the protoplasm will die. So, Mamuna, I have told you the basic concept, and ambition I have already discussed you, right? Now the next point is next heading is long distance transport of water. That is the long distance transport of the water, right? Put the and so first we will discuss the put the heading pathway of water movement. Pathway of water movement in route, right? Put the heading pathway of water movement in route. Now let me tell you, there are two pathway of water movement, right? This is very important. Your school teacher will definitely ask you this one. If it is coming in next exam, definitely they will ask you. There are two ways of water movement. So these are the epidermis is the outer wall of the root, then cortex is in between. There is one more layer called endodermis, and then pericycle, and then there will be xylem. Right. This is the sequence of the actually roots. There are two paths of movement of water in root. The first path is called actually apoplast, and the second path is actually called symplast. Apoplast in apoplastic pathway, what happened? The movement of water takes place in this pathway. Water do not enter inside the protoplasm or inside the vacuole. Actually, water move between the space between the protoplasm and cell wall. So water move in this way. This way. So in epidermis, apoplastic pathway follow. Yes, the first pathway, apoplastic pathway, it follows. Again, in cortex, yes, epiplastic pathway takes place. But when it comes to the endodermis, what happened? Apoplastic pathway is not possible. Actually, these endodermis cells, they have a Casparian strip. Casparian strip is made up of a impermeable solution or impermeable deposition of impermeable substance. Impermeable substance, right? So this impermeable substance, it do not allow movement of the water in apoplastic pathway. So in endodermis, only symplastic pathway takes place. Again, the movement takes place in pericycle and symplastic and to the xylem. through symplastic pathway. The second is symplast. In symplastic pathway, water moves inside the one protoplasm. Then between the two cells, there are some pores. These are called plasmodesmata. So with the help of plasmodesmata, water enter into protoplasm, from plasmodesmata to another protoplasm, then again plasmodesmata to another protoplasm, and through to protoplasm. So there are two pathways, apoplast and symplastic. In symplastic pathway, the water moves from one vacuole to another vacuole to another vacuole, right? But in apoplastic pathway, water moves between the protoplasm and space between the protoplasm and cell wall. In epidermis, both symplast and apoplastic pathway takes place. In cortex, both symplast and apoplastic pathway take place. But in endodermis, only symplastic pathway takes place. In pericycle, again the symplastic pathway takes place. But xylem, they are the dead cells. They do not have any protoplasm. So this is the just movement of the water through the dead cells, right? Is it clear, Mamuna?
Yes, sir.
Okay, so put the heading first, take the screenshot and put the heading apoplastic pathway. Here the water passes from root hair to xylem through the walls of cell without crossing any membrane or cytoplasm. Next point, the pathway provide least resistance. The pathway provide least resistance to movement of water. However, in endodermis, it is interrupted by impermeable Casparian strip, which is made up of lignin and suberin, Clear apoplastic pathway? Next.
Yes.
Next, Mamuna, symplastic pathway. In this pathway, water passes from a cell to another cell. In this pathway, water passes from a cell to another cell through the protoplasm. Full stop. Underline this statement. It does not enter the cell vacuole. It does not enter the cell vacuole. Full stop. The cytoplasm remains connected. The cytoplasm remains connected to adjacent cell. Cytoplasm remains connected to adjacent cell with the help of plasmodesmata. With the help of plasmodesmata. Right? In this pathway, at one more point, in this pathway, water had to pass. In this pathway, water had to pass through cell membrane. One more common point. Water flow via apoplast. Water flow via apoplast in epidermis and cortex. Full stop. It enters in symplastic pathway at endodermis. Again, one more important point for your medical examination, put a star. Mineral, nutrient also have the same pathway as that of water. Mineral nutrient also have the same pathway as that of water. Okay. Now come to the one more point. Mamana, clear? Everything clear?
Yes, sir.
Plastic pathway, right? Yes. Let me show you the actual diagram of this apoplastic and symplastic pathway. Take this screenshot if you want. This is the actually anatomy of root. Look. Again, this is the pink one is apoplastic pathway, and this blue one is symplastic pathway. These are the root here, so they follow both apoplastic symplastic pathway. This is the epidermis. This one the epidermis. Again, you will see in epidermis they follow the same pathway. Again, in epidermis both follow these are the root here. In root here, first they follow apoplastic symplastic. Then the second layer endodermis, they again follow apoplastic and symplastic. Then from here till here at this point, this is the actually cortex. In cortex, endodermis is a part of actually cortex. This one till this one. So till cortex, actually till cortex they follow the this one apoplastic and symplastic both pathway. But when it comes to this endodermis, they follow just symplastic pathway. Then they enter into xylem. So in xylem, they pass through the pressure gradient. So you can take the screenshot of this one as well. Okay. There is one more topic, that is a mycorrhiza. What is the mycorrhiza? This is also very important actually. What is the double R H I Z? Double R H I Z A. What is mycorrhiza? Mamana, have any idea? Look, what happens? Some of the plant like gymnosperms. So their roots are not capable of absorbing water that efficiently. So what happened around this root, there is a network of fungal filament. So this is the fungus. This is fungi. And this is the root. What happened? Fungi help fungi help the root to absorb water and mineral. In return, root gives them food. So this is a type of symbiotic relationship. This is a type of symbiotic relationship. This is type of symbiotic relationship between root and fungi. So symbiotic relationship between root and fungi, and this is called mycorrhiza. Clear? Namuna, is it clear? Yes. Tell me, come on. Yes, Mamina, wake up.
Yes, sir.
Is it clear? Mycorrhiza?
Yes.
So note it down. Mycorrhiza is the fungal association of roots of higher plant. Full stop. Fungi have large surface area. Full stop. The fungal hypha are specialized to absorb water and mineral. Fungal hypha are specialized to absorb water and mineral. Next, in return, root gives fungi. In return, root gives fungi. In return, root gives fungi food. Right? Example, pinus tree. Pinus. So we have studied plant classification. You know what are the pinus? So pinus, cycas, they all have mycorrhiza. Mainly pinus is the best example. They have. Clear?
Yes, sir.
And the next is again, what is guttation? One more. What is guttation? Yes, Mamuna, can you tell me what is the guttation? And let me explain. Guttation. Take a screenshot of this one.
Yes, sir.
Give me 1 second. Let me show you a diagram of guttation. Actually, for guttation, plant have a special type of the mechanism, or you can say they have a special type of force called hydrothodes. Let me first explain you what is the process of guttation, then I will tell you which help the guttation. Look, Pamela, uh, actually, there are some plant, they have small, there are some plant, basically where these plant are found, what kind of plant, I'm going to tell you everything. They have a special type of the pore at their margin. These pore at their margin, they are called hydrothode. They are just like the this one stomata, but they are permanently open. So from hydrothode, what happens at the daytime? They have high rate of transpiration. So generally, they are found at the desertic region. Desert region. So what happened in desert region? The days are very hot. So due to the hot or high temperature, transpiration is very fast, and because of this transpiration, root pressure become very high. Right. At the night time, it becomes very colder, but transpiration remains, right. The root pressure remains in the high. So because of this root pressure, the root try to push out more and more water, but as the transpiration do not takes place, so from these pores, the water droplet comes out. So water comes out in the form of the droplet and goes down. This kind of the transpiration or this kind of mode of the transpiration is actually called guttation. This is called actually guttation. So what happened? There is a special kind of the tribe or a special tribe in South Africa that is called Masai. You must have listened about those people. Have you listened about those people? Masai? Masai. So
Yes.
in Africa, there are actually large tree, wide leaves plant. So Masai people, what they do, they take the ostrich eggshell and they put this ostrich eggshell at evening time at the tip of these leaves. Throughout the night, this water keep on coming, and at the morning, this pot, or we can say this ostrich eggshell, it get filled, and they collect these water in the bucket, and the whole daytime they use this water. So this process is called guttation. For guttation, these are the hydrothode. This is the anatomy of hydrothode. Hydrothode have a pore. Again, they have a guard shell, and there are epitheme. What is the epitheme? Actually, this is the loose parenchyma tissue. Parenchyma tissue. Epitheme is important tissue. This is the intercellular spaces between the parenchyma. This is the xylem, and this is the chlorenchyma. The cells which have chloroplast. Clear?
Yes.
Ramana, 100% clear. So please note down the definition of guttation. Guttation is the process when water starts coming. Guttation is the process when water starts coming from the margin of leaves in the form of droplet. Full stop. Next point. The loss or excretion of water. The loss or excretion of water in the form of droplet from leaves and other part is called guttation. It takes place, take the screenshot of this one. It takes place in Nastotium. Nastotium is plant. In Nastotium, tomato, cucurbit, full stop. It takes place through a specialized pores. It takes place through a specialized pores called hydrothode. It takes place through a specialized pores called hydrothode. I'm gonna put a heading, next heading, water movement up a plant. Water movement up a plant. We'll start it in next class. So have a glance whatever we have studied today and tell me if you have any question, any doubt.
No, sir, no doubt.
No doubts? Okay, Memona. So see you in next class. And next class we'll study the translocation of the water from one point. Right?
Okay, sir. Thank
So
you.
bye-bye. Welcome, beta. Bye-bye. Good night. Take care.